HFSP for funding my postdoc. Big goal of neuroscience, I'm a neuroscientist, and major goal of neuroscience is to understand how our cognition works. And now the key hallmark of cognitive capabilities, be it attention, working memory, or decision-making, is the retention of information. So, for example, if you are looking for your favorite pairs of socks from your drawer, or if you're comparing, multiple bottles of wine, your brain somehow needs to keep representing the information that's no longer given to your sensory systems. But this is not a trivial task, especially given that individual neurons operate typically on a much faster time scale, forgetting the inputs they received a fraction of seconds ago. So how does the brain bridge this time scale gap, so to speak? Over the years, theoretical studies have suggested that the recurrent circuitry of these fast neurons can generate more sustained dynamics, which can then support these cognitive capabilities. However, it has remained challenging to really figure out how these recurrent circuits are actually biologically implemented, especially in the big mammalian brains. So my strategy to tackle this challenge is to study the larval zebrafish. The larval zebrafish is one of the smallest vertebrate model organisms with only 100,000 neurons in their brain, which is 1,000 times smaller than a mouse brain. In addition, there are a lot of transgenic tools to visualize and optically manipulate the activities of specific neural populations in the brain is available in zebrafish, which is particularly useful in them because of the optical accessibility of their brain due to the transparent body, which is, if you use melanophore mutants. So altogether, I think larval zebrafish offers us a unique opportunity to make tangible, real progress towards understanding the mechanisms of recurrent circuitry that supports cognition. So in my poster, I will showcase two of my recent projects along these lines. And first project looks at the visually driven stabilization behavior. So the larval zebrafish, similar to many other species, including ourselves, use the wide field visual motion as a cue that they're being dragged by the external forces, like the flow of water, and swim to counteract this motion. But this visually driven swimming has been shown to be dependent upon the history of visual motion over 10 seconds. However, the exact algorithms or function of this surprising history dependence remain unclear. So I use various behavioral manipulations as well as imaging approaches to sort of infer the purpose of this flow memory, as well as like how this time scale of this flow memory comes about. In the second project, I studied the neurons that enable fish to remember which way they're facing, just like a compass in the brain. And using a custom-built virtual reality system, I show that the conserved projection from brain regions called habenula and interpeduncular nucleus implements this flexible anchoring of their head direction memory to external landmarks. So thank you for your attention, and I'm waiting for you at poster number 28.
Thank you very much.